Hello and welcome to the Sheekey Science Show, where today I'm here in Cambridge to speak with Daniel Ives, the CEO of Shift Bioscience, where they're using machine learning to try and identify safe drugs for cellular reprogramming. So before we speak with Daniel, we're going to take a little nice tour inside the building. Cool.
Is longevity, escape velocity, right? So they're like trying to help everybody see this and get up there because it's going to be great. So yeah, Nathan sort of qualifies, I qualify. It's a little bit loose about who gets to be in this or not. It's mostly, who doesn't throw up a fuss. Anyway, so that's that, and then that's Elon Musk as he got really old.
Hi Daniel, thank you for joining me today in person and welcome to the Sheekey Science Show.
Thanks for having me.
Awesome. So yeah, it's great to actually be able to do this in person for once. And we're actually here currently in Cambridge where we both have, ties to now. And also we're somewhat related in that we both initially studied biochemistry. And so I guess my first question was, where did that interest come from? What drew you into the field of biochemistry, and then subsequently you did your PhD focus on mitochondria. And so I guess could you elaborate on what your PhD project was all about?
Sure. So the reason that I got interested in biochemistry was actually very boring, which was I was good at biology and I was good at chemistry, out of the subjects I was taking. And I just thought, well, just go with what's working. And I was very interested on sort of the biological side in particular. I thought chemistry was very challenging. There were some interesting details, but it was mostly about the biology. So biochemistry was really interesting because it was greater depth on the biology. So, I'd almost resented learning all this chemistry stuff, but when you frame it in the context of biology, it was very interesting. So I did biochemistry, and I was good at that. And then I went all the way to masters, and around the time I needed to decide what I was going to do after that, Aubrey de Grey, who's a Cambridge geneticist, had written a book called Ending Aging, and it was really like a call to arms. It was like that aging isn't an inevitable process. You and me, we're not like a rock subject to entropy, right? We don't just turn to dust, right? We can externalize entropy. There's no reason why we shouldn't keep ourselves going the way you could keep a vehicle going, right? Like classic cars, you can keep them going. And it was captivating, this book. It's really sort of caught my attention. And after reading that, I was like, look, this is the way that I can really leverage my skills. Let's focus on this. And I think one of the top, types of age-linked damage listed in the book was mutations in the mitochondrial genome. That was the hardest one to crack, so I was like, well, I'll do that. And there was a project in so the mitochondrial biology unit in Cambridge, actually sponsored by Aubrey. And I applied to that project, but by the time I showed up, it was too late. So basically the first student had encountered technical difficulties. They'd had to discontinue the project. Okay. I didn't really have a plan B. I just wanted to do that project. But sort of in my morning period, I just found other projects in the lab which seeked to address the same problem, which is as you get older, mutations in the mitochondrial genome accumulate. And there was an alternative way to sort of try and fix this problem, and that's where I got started with my PhD. And it went well. So there's a publication came out recently, and this work started way back in 2009, so it just shows you how long it can take.
Yeah.
I think it was three or four postdocs had to sort of come in and lead it. So I wouldn't say abandon ship, but I had to go and do my own thing. I wanted to basically work in aging, and the lab wanted to work on rare diseases. But there was a few postdocs, and that is now published. And it's funny because the publication is almost the palatable version of events, which is actually, I did a very messy and I think more interesting thing, and we discovered that we could actually use small molecules to get rid of mitochondrial DNA mutations, right? But it's still a lot of black box. So these poor postdocs had to try and reverse engineer this black box so it would be fit for publication, because nobody wanted to know about the messy way we'd create this black box and found this molecule. But once there was a bit more story around it and mechanism, that was more palatable. I think in the early days I was actually accused of just arriving at this sort of black box by pure luck. Someone was like, well, you could have just got lucky, and I couldn't disprove that. But I'd actually worked hard on the bioinformatics, sort of self-taught gene set enrichment analysis. There's something called ingenuity pathway analysis that I sort of took on, and we arrived at a prediction of what was going on, and it worked. So maybe I did get lucky, but I feel like I put in a lot of work as well. So it doesn't really matter. We found out something that worked.
So I guess it's worth mentioning then that, so we obviously have our DNA in our nucleus, which has our genetic information, but it's also worth pointing out that we do have these separate mitochondria that also have their own DNA. And I think one of the interesting things about mitochondrial DNA is the fact that we have multiple copies of mitochondria in a cell. And so some of them might get mutations and some of them might not. And so you get this thing called like heteroplasmy. Yeah. But then the other thing with mitochondria, right, they only code for 13 genes, is that right? Or around 13 different genes. So they're quite small compared to our whole nuclear genome. So like, what is the kind of arguments for mitochondrial mutations actually being important to the aging process?
So the most provocative finding was that if you turn up these mutations, like I think it's like 2,500 times higher than you would experience physiologically, at least for a mouse, you get a premature aging phenotype. So it's called the mutator mouse. Mm-hmm. So basically it just exhibits all the features of aging, all the phenotypes of aging. And I think it was a Nature front page. It was like aging solved, like just very presumptuously, it was like aging solved. But there's lots of ways to create a premature aging phenotype, because like mutant nuclear envelope proteins, like lamins, there's a mutant lamin, creates an aging phenotype. There's lots of progeroid models like double strand break repair deficiencies create a kind of aging phenotype. So, I guess there was all those things going on, but because these mutations accumulate with age, and when you turn them up, you create an aging phenotype, it was sort of very provocative at the time. I think it's like 2004.
Okay.
So I guess, I'd almost made my bed at that stage where I was at the mitochondrial biology unit because I was interested in aging, and that's why I was working on mitochondria. That was my world, and I was like, well, it's almost confirmation bias, right? It's like, mutator mouse. And I didn't know about the aging clock at this stage, right? This sort of relatively unbiased way to measure aging. So I guess in the absence of that, it's like everybody was in contention. It's like, well, it might be the mutations, or it might be senescent cells, or it might be autophagy. And so I was just, going a million miles an hour with, my route. But we were able to find sort of molecules that could basically get rid of these damage types. So the mitochondrial genetic system is very interesting. Like thousands of copies potentially of a mitochondrial genome in the cell versus just 2 copies of a gene, right?
Exactly,
the diploid nucleus. And it's almost like having 2 operating systems, because in the nucleus, if you only got 2 copies and one goes wrong, that's a big deal. That's like 50% of the information gone. But in the mitochondrial system, you can just get rid of one copy. You can sort of by autophagy, get rid of one of the mitochondria, just clear it out, and then they can reproduce and repopulate. So it's actually more like an ecosystem, right? You've got all these mitochondria sort of coexisting with each other, and what tends to happen is one of the mitochondria gets a mutation in its genome. That genome gets some sort of advantage, So the genome creates a mitochondria which isn't as good, right? It's not as functional, but it replicates faster, or it's somehow it's able to replicate faster, and then it just wipes out the cell. So when you look at aged tissue and you see a cell that's deficient in energy production, which is sort of a sign that the mitochondria are going bad, you go and look in that cell, and it's a point mutation in the mitochondrial genome, but in every single one of those 1,000 genomes. So it's like, there was some event and then that was like an internal cancer and it wiped out that cell. And you go to a different cell in an aged tissue, there's a different point mutation which is wiped out that cell. So it's this sort of instability in that genetic system, right? It's sort of waiting to happen sooner or later, like in a probabilistic way. So the mitochondria basically, they have all these copies, which is great for redundancy, but then they have this fragility, which is like if one copy takes hold, it's a big problem, and there's no repair, relatively little repair there. So.
Exactly, yeah, because I think when I was taught about it in undergrad, there are certain like mitochondrial associated diseases, and they occur with age because it takes time for like the mutated copy of the mitochondria to then obviously kind of like take over the cell in terms of the copy number compared to like the wild type equivalents.
Yeah, and there are surveillance systems in the cell. So these mutant genomes, they just hide below the surface of those surveillance systems. So they're not so dysfunctional that the surveillance systems pick them up easily. There's like pink one parking pathway.
Mitophagy stuff.
Yeah, it's all mitophagy. Okay. So basically, if you have a massively depolarized mitochondria, this just scoops it up like immediately. But if you're on this sort of middle ground or grey zone where you're still making a bit of energy but not as much as you should be, you can sort of be a bad genome and then over time like slowly take over. It's like a sort of insidious takeover. Yeah.
And so you spoke about this recent publication that I believe came out literally at the end of last year. And so in it you describe 2-deoxy-D-glucose as being one of these compounds that can like somehow bias the cell to get rid of the mutated copies. So like, is that something we should be really excited about as in a potential therapeutic, or what are your thoughts on it?
Yeah, so 2-deoxy-glucose is a double-edged sword. So the way we even like got to 2-deoxy-glucose was sort of interesting. So basically there's a cancer cell line called A549 that spontaneously figured out how to get rid of damaged mitochondrial genomes. Yeah, so if you populate this cell type with a mutant genome and a wild type genome over time, which is like a period of months, it will get rid of the mutant genome. Like repeatedly and reliably, it will do this, which is the opposite to all the other cell lines surveyed. So like other cell lines like 143B, osteosarcoma, there's some other ones. They all do the opposite, which is if you put a mixture of a mutant genome and a wild type genome, the mutant will increase over time. And this will happen in typical cells as well, otherwise these things wouldn't persist. So the way that I found 2-deoxy-glucose was that I took transcriptomes from the NCBI-GEO, which is a big public database full of transcriptomes. So I took them for A549. There's loads of them, right? Because loads of people use these cells for different experiments. You just take the control.
Yeah.
So there were like 30 transcriptomes for that, and then I took equivalent number of transcriptomes for the other cell types to do the opposite. And then I just did differential expression, and then I did gene set enrichment analysis. And that was actually just with gene set enrichment analysis that we saw that the unfolded protein response was really upregulated in A549 versus the other cell types. And then it was no more complicated than this. I was like, just bought a bunch of stuff off Sigma that activated the unfolded protein response. 2-deoxy-glucose was one of them, but there were plenty of other things I tested. But it was only 2-deoxy-glucose that basically recapitulated what the A549 cell line was doing. Like there were actually things that activated this unfolded protein response, which didn't reduce the mutations. Yeah, so it's not as simple as unfolded protein response equals getting rid of these mutations. But something about 2-deoxy-glucose is, this is why people are like, oh, you got lucky, and they might be right, but it does work. And the moment you get something to work, you can use that as a way to now we can induce it on demand. We can start studying the different features of the cell, start reverse engineering the process, right? Like we just induced the phenomenon that we're interested in, and let's see what different pathways in the cell are doing. And that was the reverse engineering project that followed, right? Which I wasn't sort of deeply involved in because I was doing stuff with Shift and aging. It was once we knew the utility existed, right, of these molecules. For me, it was always about aging. Let's figure out what's going on here under the black box, and this is the other side of 2-deoxy-glucose. So a long time ago, I think it was NIH investigators actually tried to use 2-deoxy-glucose as a caloric restriction mimetic, right? So because it's glucose.
Yeah.
So you just change it slightly, and it fools the cell into thinking. It basically blocks glycolysis.
Yeah, the first step, right?
So basically, the cell thinks that it's just run out of glucose, and it sort of does all these things. So they were hoping this would be like the caloric restriction mimetic, like we don't all have to be hungry all the time to get the benefits. You just take 2-deoxyglucose. And they gave it to rats, but unfortunately in the rats, they got cardiac toxicity. So they started getting vascularization of the heart tissue, and they started dropping dead before the wild type animals dropped dead.
Okay.
Yeah, so I think it was actually, my working memory on this is a bit hazy. And then they started looking at artificial sources of 2-deoxyglucose versus natural sources, like was there some chemical in the preparation that was causing this? But the short story was there was basically the cardiac tissue in particular takes in a lot of glucose, so therefore it enriches 2-deoxyglucose to much higher levels than any of the other tissues experience. And actually you sort of need a Goldilocks zone of glycolysis. You don't want to shut down the whole thing in quite a high energy tissue. But that was the idea for caloric restriction. But the reason we used it was completely different. Like we were just trying to emulate this weird cell line called A549, to activate the unfolded protein response. But there is a light at the end of the tunnel, which is you can add an additional compound to sort of get around some of these issues with 2-deoxyglucose. And I should know the paper better, but there's another compound you can add, and I think there's a quite a well advanced initiative now to run a clinical trial with 2-deoxyglucose, basically treating patients that have a pathogenic mitochondrial genome that basically causes a rare disease called MELAS.
Okay.
So you inherit this from your mum because your mum gives you your mitochondria. So they're going to try and use 2-deoxyglucose and this sort of safety enhancer at the same time. So that's great. So that's actually gone somewhere. But I'm not super involved in that anymore because we went a different direction.
Exactly. Yeah. So that's really interesting. And I didn't know that much about 2-deoxyglucose. That was really interesting to hear. And having worked with A549 cells as well, that's also quite interesting. I didn't know that about those cells.
Yeah, like what is it about the evolutionary history of that cell line? Yeah, because it's a lung cell line.
It's lung,
so it's lots of oxygen. So if you've got a lot of oxygen, and you can use your mitochondria, right? Maybe that's a selective advantage. I don't know. Like if you've got functional mitochondria that allows you to grow faster as a tumor, I don't know. I can only speculate on that, but it is intriguing the fact that it's actually unique. Just this lung cancer cell line has found out a way to mix up its genes in such a way that it can enrich like the wild type mitochondria at the detriment of the mutant, where other cell lines don't do that.
Yeah, for sure. But I guess we should probably shift over to Shift, as you've really brought it up in conversation. And so you've, at this time you've finished your PhD and now you're interested in mitochondria still and its links with aging. So how did you go from that position to starting Shift Bioscience?
Yeah, so whilst I was in the academic laboratory, like looking at 2-deoxyglucose, I knew about the mutator mouse. Yeah, so like the mutator mouse was this premature aging model. That was caused by mitochondrial DNA mutation. So the models I were using were basically rare disease models. So it was a MELAS mutation, it's a point mutation that causes rare disease. And we use this to see, does 2-deoxyglucose reduce the level of mutation or increase it? And when we got something working there, for me, that was the moment. It's like we found a technology that can help aging. And I just wanted to do the equivalent experiment in mutator mice. But these are very expensive experiments. Like you've got to get a bunch of mice, you've got to figure out how to dose them properly and all these things. So it was very risky, it was high budget, it just wasn't the sort of thing that the laboratory I was working at could bear. It was really for me to go that direction. So for various reasons, my postdoc was cut short by 1 month, just 1 month. Okay. But I got a fantastic redundancy payment. It was like £10,000, and you don't pay tax on it either. So it's just nice. £10,000. So I wasn't able to do mutator mouse experiments with that, but I was able to at least look in a second system. I think it was like a Parkinson's fibroblast cell line. So we had an inclination that there'd be a mutation, right, in the mitochondrial genome of a Parkinson's cell line, which in hindsight was actually quite a high risk. That hypothesis was nowhere near assured. We actually found in the only available idiopathic Parkinson's cell line, we found a point mutation in the mitochondrial genome. I think it was like ND5. And then we were like, oh wow! So we've got this thing working in the MELAS setting. will it work in a general sense, right? Will it work for any mutation, not just MELAS? So we basically, we tested, I think it was 2-deoxyglucose first, in this Parkinson's cell line. Unfortunately, it killed the cell line, right? So it's a fibroblast from a very old individual with a disease, right? So 2-deoxyglucose is pretty severe anyway. So anyway, it killed the cell line. And then we had to basically go on a search for structurally similar compounds to 2-deoxyglucose. We were like, okay, what is it about 2-deoxyglucose that's interesting? Well, let's stay within the structural realm. So we found some compounds that were structurally similar, but didn't have this extreme viability issue. And so one of those compounds, called SP200, we treated these Parkinson's fibroblasts, and we got rid of this mutation. Yeah, so whatever this activity was, it wasn't specific for MELAS. We could look at, we could deal with that mutation. We could deal with that mutation. And the moment we knew that we could, it was independent of the type of mutation. That was the de-risking step before the mutator mouse. So the mutator mouse is just mutations everywhere on this genome. And so we needed to know that this was a general activity against mutations, not just specific mutations. And then we basically just went around asking people for money. So I think my dad put in a bit of money, my uncle put in a bit of money, we got some donations. I asked my girlfriend at the time if she'd give me her life savings, and she said no, which was the correct answer. She said you should talk to an investor who can afford to lose this money. So it was a very high risk project. At the time I thought, we were on to, this was going to be it. But she said to go to an investor, and because I've got, my dad was a serial entrepreneur, right? So quite a long time ago, he has a network in Cambridge. I was very fortunate. We could just, he could see what was going on, right? He was getting interested because he's got a biochemistry degree as well. But he went into like mobile technology, right? He didn't go into life science. But he could see this was getting interesting. And so we were actually able to connect with Jonathan Milner, who in Cambridge, UK, he's a life science angel here. So he founded the company Abcam, which was a unicorn. And then he obviously did well, and he's angel investing off the back of that. And we went to talk to Jonathan about this, and he said, this is too early, I'm not ready for this yet. But if you manage to do anything with the small amount of extra money you're about to spend, because we did have a little bit of extra money, he said, I'd like first refusal, if this becomes a company. So we went back, actually made a bit of progress, and he put in the first chunk of money. Like, we actually established Shift, right, in 2017 at that stage. So that's how Shift started. I'm going to stop there just in case, because I'll just keep going.
Cool. Yeah. So initially you had the idea of focusing on mitochondria still as being the focus point for finding some kind of therapeutic. But then at some point you've now shifted gears more to just like cellular reprogramming and looking at using machine learning and gene expression data to try and find an answer instead. So pardon the pun, but how did you shift your focus between these two different areas?
Yeah, so just to make sure there's no confusion, the term shift doesn't come from our propensity to pivot as a company. So it actually comes from the mitochondrial DNA heteroplasmy, which is the coexistence of two populations of mitochondria, each one with a mutant genome, one with a wild type genome. It's called heteroplasmy shift when you're able to change those proportions. So the A549 shifts to wild type, another cell line shift above. But for various reasons, Shift seems to work in a lot of different contexts.
It does, yeah.
I don't know, we got a driver clock. It does seem that there's no need to change it right now. It just seems to have earned its place at the table. So when we set up with Jonathan Milner, I was absolutely adamant, right? This was the solution. This was the point of maximum leverage. This is how we're going to solve aging. Just have to get rid of these mitochondrial DNA mutations. But very quickly, it was actually Jonathan that, put this across my desk. He was like, "Have you heard of the aging clock?" Because he sort of still reads all the journals, right? You see them on his desk. He said, "Have you heard of the aging clock?" And I was like, "What is the aging clock?" And this was 2017, so it wasn't like it had just come out. It was like 4 years ago, and 4 years had passed. And the moment we found out about the aging clock, it was like, this is amazing, right? Because now you've got the ability to measure aging. So for us, we had a very narrow hypothesis, right? Mitochondrial DNA mutations are a big cause of aging, or one of the biggest causes. if we reverse that, we're going to reverse a lot of the process. So let's qualify that using the clock, because the clock is as close as we can get to a robust compass of, like what is aging, what isn't aging.
And to be clear here, you're talking about the epigenetic...
Epigenetic aging clock, yeah. So Steve Horvath's epigenetic aging clock. But the experiment we wanted to run was using mouse samples. So we basically did the mutator mouse experiment, and excitingly, we showed that we could slow down the visible aging phenotypes in this mice. So it was like, oh wow, it actually works in the mouse. Like, I think I was advised by my external PhD examiner that this was don't do this experiment, it's high risk, but that's almost, this is the type of, you set up a startup to do these things, right? But I did it, and we actually affected these aging phenotypes. Like, oh wow! So it's actually amazing to, like, you've got this mouse experiment, and you get updates, and you see that the treatment, right? So you've just had a hypothesis, right? And you see that treatment, these mice are better off. It's actually pretty, like, that's quite an amazing feeling.
Yeah.
And, like, we didn't know that this wasn't the be all and end all of aging at the time. So it was even, it was like, oh wow, like this reinforces that we're doing the right thing. But we wanted to make sure that we weren't accused of just affecting aging in this particular mouse model, because people always complain, right? Get it to work in a mouse, doesn't really mean anything. It's good for the mouse, but we're not mice, right? So, like, are we doing all this for the mice? No, we're not. It's for us. So we wanted to use an aging clock to show that not only was this mouse model affected by this therapeutic, but the clock was affected, and therefore physiological aging was affected, right? Because the clock represents physiological aging. So Wolf Reich, who's actually a local academic at the Weber Institute, and now he's moving into the sort of Altos labs, he came up with the first multi-tissue mouse clock, which allowed us to basically measure the clock in our mouse samples. But we had to embed somebody in his lab because he didn't have any spare hands. We couldn't just send him samples, right? So he said, why don't you send one of your people, right, to learn what we're doing, just minimize the amount of bandwidth required from his side. So send them, and they can learn it, and they can do the clock measurements. And we did this. So we got a really interesting result from this experiment. So on the one hand, it was good. On the other hand, it was the beginning of the end for this approach. So we did slow down the clock by 50% in this mouse model. And on the back of that, we actually set up a follow-on experiment in aged B6J mice, so aged physiological mice. So you can actually buy like 78-week-old mice from the Jackson laboratory just off the shelf. They maintain a cohort. And so we repeated this experiment in these aged mice. But whilst that was going on, because it still takes a long time, we showed that the mutator mouse didn't show any acceleration of the clock at all, right? So the clock basically said that these mice that visibly look very much older than they should be, according to the clock, they were exactly the age, like exactly their chronological age. So even though we'd slowed the clock down in this mouse model didn't show acceleration of aging, right? Which is, that's the whole, if the mutator mouse has way more mutations, it should have a much higher clock reading. So it turned out later, like when we did the wild type mouse experiment, that we didn't slow the clock down in the wild type mice. It was only in the mutator mice we could slow the clock down. But the major finding was that the mutator mouse wasn't accelerated aging according to what the clock represents. So it wasn't the acceleration of physiological aging.
Okay.
There's an acceleration of the aging phenotype, but there wasn't an acceleration of the clock. And we really put a lot of faith in the clock. Like, for us, it was like, look, we have to affect the clock first, and then we can do other things, and that's great. But to not affect the clock is just too risky down the line. Like, we want to be focusing on the absolute problem that you and me have, right?
I see.
Not something in a mouse. So for us, there was a lot of soul searching, also known as pivot point or existential crisis. There's many names for it, and you have to ask yourself serious questions, which is like, why am I doing this? So we had a question. There's a sort of fork in the road. Do we continue to develop this type of therapeutic, which ultimately belongs in rare disease patients? Like that's where it was born, and that's where it had the most utility. Or do we stay true to the mission here, which was the initial mission of Shift, solve aging. I know people are a bit funny about the language now, but let's just solve aging. And for us, it was always about that. That's where we get the energy. It's like, it'd be amazing if we could bring age under our control. It was up to us how much we let ourselves unravel. that just changes everything. It's like the equation just changes for everything. So we tried to like learn the lesson from this result, because actually using a clock was, we were trying to face the music, right? It's actually a brave step. It's sometimes it's quite hard to stare the truth in the face.
I can imagine.
So we wanted to learn the lesson from this, and actually the clock had told us a valuable lesson, which was we're not as clever as we think we are. Like we like to think we're clever, especially at Cambridge University. Like you sort of all of the history, right? And I got to Cambridge. I remember my parents saying, "Oh my god." It's like, "Oh, you're so clever." But no, you're not. So nature's got a lot of mysteries that are it's not always brain power. So the lesson that we'd experienced was like we'd just been able to fail fast, right? We're no longer the walking dead.
That is the best way to fail.
Yeah, so we're no longer the walking dead, because the clock has said, all right, it's given us some valuable information. This isn't the way forwards. But we'd actually use this clock in a very narrow sense. So once you've got the ability to measure aging, the potential of that isn't just to ask one question at a time. The full potential of the clock is to ask all of the questions, right? And so, like biologically, it's something like a functional screen for aging or a CRISPR screen for aging. So, you can knock out gene 1 in the genome, see what it does to aging, knock out number 2, gene 2 in the genome, see what it does to aging, go through every one of the 20,000 genes and assign a relationship to aging. And if you do that, you can let the biology do the talking, right? Let the biology tell you where to focus and say start building as far as drug targets are concerned. So for us, that was the lesson from the clock, right? And that was the product of the pivot. So it was like, well, let's focus on clocks. And we had a particular technical problem we needed to solve to do this CRISPR screen. So we could have done a CRISPR screen with the existing methylation clock, the Horvath clock. But every clock measurement actually requires a large number of cells in a dish. It's like millions of cells for one clock measurement. Okay. So it's actually like a 10 centimeter dish for one clock measurement. So you actually need like 20,000 dishes, right, to do a CRISPR screen. It was just sort of unmanageable from our standpoint. I mean, maybe you could do it like a human genome project type thing. But for us, it was just, that was unmanageable. However, 10x Genomics had launched a machine called the Chromium, which allowed you to do single cell CRISPR and simultaneously get a transcriptome from each cell. So you could knock out one gene in a cell, get a transcriptome, knock out another gene in a cell, get a transcriptome. And this has actually offered a path to a CRISPR screen for aging, because though they weren't as well used and not as celebrated, you could create aging clocks from gene expression or the transcriptome. So I think Alex Z from In Silico had created a transcriptome clock. Others had created transcriptome clocks, but they were less accurate. They were always tissue specific. They sort of became the B team, and then the methylation clocks. They always got better, and like multiple species, mortality, grim age, those sorts of things. Like that really became the center of focus. But we wanted to do a CRISPR screen for aging, and therefore gene expression based clocks became very interesting very quickly. So if we could create a single cell aging clock based on the transcriptome, we could do a CRISPR screen for aging and actually find the aging biology. So like it's almost the key to the biology then became a gene based clock, a single cell, because it had to work on this single cell CRISPR screen. And fortunately for us, Tony Whiskeret at Stanford had deposited this amazing data set called the tabula muracenis. So for those that aren't familiar, it's single cell transcriptome of the mouse, like every single cell from the mouse at the transcriptome level over an aging time course. So it's like, I think it was like $2 or $3 million, right, to fund all of this, and it was made publicly available. And we could use that single cell transcriptome data to try and create clocks. And this is where Brendan Swain comes in. So Brendan was originally an intern at Shift, I think in 2018. Like it was actually relatively recently after we started. And we initially got interested in clocks because we were using clocks to audit our hypothesis. We were also trying to share it with people because the clocks were not familiar to people, right? And everybody else was trying to figure out how to use clocks, and we were using them for our own purposes because we wanted to sort of do the therapy side. But yeah, he got interested in clocks, started playing around with the Horvath linear regression, all the code and stuff like that. And then we simultaneously got interested in this tabula muracenis, and we almost combined the two, right? Like let's just take this sort of off the shelf machine learning method, right, which is generating clocks. It's just supervised machine learning, and try and make this work with single cell transcriptome data. And actually, Brendan he's self-taught, so he's a biologist, right? But he's self-taught machine learning to try and crack these problems. And we did crack this problem. We created a single cell aging clock with comparable accuracy to methylation clocks. So there was hardly a deficit, but it was single cell. So I think this was. I don't want to write the history books, right? But I think we were first. Like having a single cell aging clock, and it's not probabilistic based. It's like we actually get really robust measurements like at a single cell level, and we were like, wow, we've done it. And I think the secret sauce. And I've been told to remain stealthy on the details, right? Because everybody's migrated to sort of commercial entities, but it was basically noise reduction. Gene expression is noisy. If you can get rid of the noise, you can start seeing biology properly, like in focus. So that was mostly it. And then we were like, wow, we've got this clock. Like, let's do the CRISPR screen. It's like, wow, all of a sudden, we're ready to do this CRISPR screen. But it was actually a mathematician that pointed out something that should have been blindingly obvious to us as biologists, which was the biology sitting inside this clock. Because it was built out of genes, there was a lot of biology to look at, because genes are easy, it's easy to experimentally interrogate genes, relatively easy, right? You can overexpress a gene, knock out a gene, see what the impact is. Many PhD students work on genes and proteins, right? They get lumped with them or they enjoy working on them. But there's a lot of work that's been done on genes. Unlike methylation sites, right? It's very hard to experimentally interrogate a single methylation site and show that it's linked to this and that. Like, the tools don't exist experimentally. That they're starting to be developed, right? There are methylation editing, but it's still the experimental stage. But genes are fantastic. So the mathematician basically pointed out, oh look, sitting inside your clock are aging biology. Like, there's genes, it's like there's genes that are known aging biology. So there were mitochondrial genes, there were ribosomal genes. So there's a ribosomal clock, like on its own creates a clock, from Bernardo Lemos at Harvard. So, and there was a gene that on its own creates progeria in humans, right, sitting inside our aging clock. And we realised that the more interesting thing about this clock wasn't using it as a readout for a CRISPR screen. It was the cogs inside the clock, which is these genes, which is, it's like a window into the aging biology. And then all of a sudden we were like, that's actually more interesting, because you've arrived at this collection of genes without going through a constriction and combinatorial space. So when you do a CRISPR screen, you're actually just knocking things out one by one. Yeah. So your combinatorial space is actually very limited. But with machine learning, you have a completely open view of, like, it could be hundreds of genes or it could be just a few. So you can throw a net to everything, and the machine learning basically brings in, it's a composite of a set of genes that correlate. And we realised here's a potential window into the biology that is not combinatorially constrained. And we've done it. We don't have to spend a penny. The CRISPR screen is still going to be expensive. And so we were like, well, let's take what we think is this aging pathway and flip it into reverse in an age system and see what happens. And around this time, I think there was a lot of noise about cellular reprogramming. So I think we were aware of Tamir Chandra, he deserves more credit, right? Because he took two things which were breakthroughs, which was the Yamanaka factors and the epigenetic aging clock, and he put them together, and he showed, that Yamanaka factors can reset the clock all the way down to zero. Like, just within a few weeks, right, they can bring it down to zero. So he qualified cellular reprogramming as the ultimate rejuvenation paradigm. That's his contribution. Oh God, I lost my thread.
That's all right, I can intervene. So far you've generated this clock based on cellular gene expression using machine learning. And so initially you developed it based on mouse data. And so I guess two initial questions from my side is, firstly, are you still working in mouse cell lines at the moment? And also, how does it take into account, as we've noticed before, different tissue types? And how is, like, aging isn't just one tissue aging, it's different organs aging, maybe at different rates. And does that mean you have multiple different clocks for different tissues, or have you been able to just make one universal, like, gene expression clock?
Yeah, so we started in mice because it was publicly available data, and we could just go everywhere in that data set. Like, we could ask these types of questions, like, can you make clocks for everything, or is it still very tissue specific? It's a bit of a middle ground. So without the special sauce, right? You can create a clock for a particular cell type in a particular tissue that's accurate, right? But it just falls apart, right? Even in the same cell type in a different tissue, it falls apart.
Yeah.
But if you add the secret sauce, you can actually make that clock work pretty much as well in the same cell type in a different tissue. So we're able to cross that bridge, which is like the same cell type but in a different tissue. We can make a clock that works. So it's not multi-tissue, it's like multi-cell type, multi-tissue, single cell type, multi-tissue. We won't really be able to, we couldn't really create multi-tissue clocks with gene expression data, right? We never tried very hard because we were more focused on something else which sort of came into play. So that's as far as we got as far as multi-tissue is concerned. But then there are human data sets. So there's single cell transcriptomics for fibroblasts of different ages. So we looked in there, and it does look pretty similar. I think it's Basel commented that even though age-linked diseases, like say you try something in a mouse model of an age-linked disease, it doesn't translate very well. Biology seems to be remarkably conserved between species, like much more conserved than the age-linked diseases are.
Okay.
So it doesn't mean that we're exactly the same as a mouse, but we're more like a mouse as far as aging is concerned than Alzheimer's is concerned or something like that. But human cells are our focus, right? We're not doing this for the mice. So once we got something to work in mice, it was like human cells immediately. So there was some human fibroblasts. We looked at an aging time course in human fibroblasts, pretty much spat out the same sort of thing, with a few exceptions. But then we realised that aging is just a black box, and we've created a method that we can point at a black box, and it creates a prediction of the biology, right, that's operating inside that black box. There was almost as if not more exciting black box called cellular reprogramming. So this is you can rejuvenate with Yamanaka factors. So these factors originally discovered for their ability to take an adult cell, make it into a stem cell, but Mitochondria showed that you actually reset the clock in the process. So this whole thing's a black box with a few exceptions. So we just pointed the method at this black box, and it creates a prediction of the biology, right? And it's actually fascinating. There's actually an overlap between the aging side and the cellular reprogramming side. And ironically, right, mitochondria sick. That's where I came from originally. And it's almost I'm trying to transcend the hypothesis-based approach. But they crop up, and I mean that's enough. That's enough to say. But I guess that is how we arrived at what our prediction of the pathway is. And for us now is really here's a prediction of the pathway. Let's rigorously test this experimentally, which is like let the cells tell us if we're onto something, or if it doesn't work, that's valuable information. We can change the way we model things to make sure we actually get some information. But I guess for us, the moment we can rejuvenate without Yamanaka factors, it doesn't matter whether it's still slightly dangerous. We can rejuvenate without Yamanaka factors. There's basically a triangulation we can do where we just sort of reel it in over a period of time. There's a certain process we're putting together, which is that, that's where I have to sort of end the details. But that's it. We've got this prediction. Might work, might not work the first time, but we've got a few ways to try and make it work. But the moment we get something that isn't Yamanaka factors, triangulate from different paradigms, and it gets very interesting.
So one thing I guess I would want you to elaborate a bit on is why not Yamanaka factors, and I think it comes down to the idea about safety, and I guess that could lead on to a conversation of the cellular reprogramming, but the different extents to which you can reprogram a cell such that it doesn't fully lose its identity and could become potentially cancerous. And I guess on from that, if you want to discuss about how you foresee the work you're doing at Shift Bioscience being applied therapeutically, what would be the kind of vision down the line as to how you would use this information and be using it to treat aging?
Sure. So sort of speaking about the Yamanaka factors and the safety dimension. So I think it's useful that there are people, there's companies that are trying to take something that works and do something useful with it. The concern for me, which is, that's just our position, is that the safety window is just too narrow at the moment. So, we've got this black box, which is it's an amazing black box, right? We can rejuvenate with this black box, but it remains a black box, and these are quite powerful factors, right? They affect cell identity, and we don't really want to be doing too much in a cell, right? In living people, right? We want to be very precise about what we're doing. We don't want to be activating all these other things. We don't want to have these off-target effects. So for us, it just seems obvious. If you've got a black box and you want to increase the safety window, let's figure out what the rejuvenation pathway is inside it, because that's the best way to be precise. We find out what that particular pathway is, target that specifically, just have a greater safety window. So I guess in the long term, we're not running these huge risks. If these ultimately getting a drug into people's hands, where you can say reset your biological age back to prime of life, that's a very long road. It's going to take a lot of money. These are very simple things we can do early. I mean, they're not simple, but we do have tools that allow us to give ourselves the best possible chance of finding out what that pathway is and removing the safety problem. So, I'm sure there is a window for using Yamanaka factors, but I think it's quite narrow. And by all means, somebody prove me wrong, because it's fantastic, right? We can get the rejuvenation benefit without the nasty side effects, but it just seems obvious. Here's a black box which is dangerous. Let's look inside of it and get rid of the dangerous bit. And I guess maybe we're procrastinating a bit about it, but I think that is, for us, that's the long-term project. So that's speaking about the safety side, the Yamanaka factors, and then I think your second question was sort of like, how do you envision this?
Exactly, yeah.
So my favourite person at the moment is called Nal Barafian. So he heads up Flagship Pioneering, which was the company that created Moderna. So it's like a company creation sort of venture capital firm. And he's got a great quote, which is, he says that what just happened to the world is the combination of the fast pandemic of COVID-19 with the slow pandemic of chronic disease. So it's a collision of those two, right, which is really the big problem. So there's a solution for the fast pandemic, which is the vaccines, right? All of a sudden we have these vaccines in quite a short amount of time. But there is no solution for the slow pandemic of chronic disease. And what he's really talking about is aging. That's the cradle of these chronic diseases. So the interesting thing is, if you can crack that problem, right, the slow pandemic, it actually cracks the other problem, which is the fast pandemic. Because the concern is the severe reactions to COVID, right? Young people probabilistically just don't get as many severe reactions. If you're much younger, you don't really suffer COVID, right? It just comes and it goes. Yeah. With exceptions, but so if you could actually solve the slow pandemic, you'd end up solving both of them. And the problem is, if you don't solve the slow pandemic, everything starts to feel like a pandemic. Like, imagine we all start aging, a larger proportion of us become aged, which is the trajectory at the moment. Every little thing is we're going to become more and more dangerous the older that you get. Yeah. So things that we don't call pandemic now will become. The effects will be the same as what happened just now. So Nal Barafian's recognised this. Someone needs to deal with the slow pandemic. But I think you've recognised this. We recognise this. He's just reframing it in a really useful way. And something he does, which is very useful, it's called future backwards planning. So here's this place we want to go, and how do we get there, right? As opposed to let's just do something proximal to what we're doing, and then just do something proximal again. Like that's not necessarily where breakthroughs come from. It's almost like here's where we want to go. How do we pull ourselves into this reality? So anyway, it's a useful mindset. And if we start with where we want to go, where do we want to go with Shift, right? Like what is the ultimate goal? Imagine in the future, it's a little bit like COVID-19, right? But it's like this parallel system. So with COVID-19, you do these lateral flow tests. Just make sure you haven't got COVID. Well, that's fine, you haven't got it. Or you take mRNA vaccines as well to make sure you don't get it. So imagine in the future you have an equivalent test, and I guess you take a swab and you drop those droplets on that little bit of sponge. It goes up. Far too much detail. But instead of saying whether you got COVID or not, it says, oh, you're in the danger zone. Like by danger zone, you're within 5 years of a cardiac event, right, or 5 years within early signs of dementia. So you get that result, and all of a sudden you go and get a mRNA vaccine. So we anticipate it'll be a combinatorial mRNA therapeutic targeting a set of say 3 to 4 safe rejuvenation genes. So you go and get your again, it's just an injection like the vaccines, and you take a follow-on test and you see, are you out of the danger zone? Maybe you're not. Maybe you got too far into the danger zone and you need something that's a bit more specialized, which is, deals with the aging, but also deals with the disease-specific nature of what you've got. So maybe you're more towards Alzheimer's, more towards heart disease. So there's a slightly more tailored version, which is, like rejuvenation plus, a reversal of whatever that disease is. So, but you can see that's the sort of future you could imagine. And then, how do we get there, right? That's the key thing, right? Like future backwards. So in the very short term, what we're just trying to do is like find out what this rejuvenation pathway is, right, inside of cellular reprogramming. And this rejuvenation paradigm is just like the most powerful out of the ones that are out there. It's dangerous as well, it's a double-edged sword. It's powerful, but it brings dangers. But say we find this pathway, that's step one, but then we need to find the key nodes, right? Because for a therapeutic, you can't be targeting 30 genes simultaneously. It's just too much complexity, too many things. You want something like a set of 3 to 4, like Yamanaka factors, like there's 3 or 4 people can use those and induce those. So we want to find, from an intervention perspective, a set of 3 to 4 genes, and we think it's going to be a combinatorial RNA therapeutic. And there's a question over which tissue you target first. And we've got various ideas about this, but I think there's various conversations about where to go. I think the brain's a very good place to go first because it's relatively satellite to alternative rejuvenation paradigms. So, for instance, young blood. The idea of a change in blood composition, which is inherently more translatable than what we're talking about here. So, just changing blood plasma composition. There's a fascinating study. I think Steve Horvath and Harold Katcher, where they show that you can basically rejuvenate the clock by over 50% in lots of different tissues, but the brain stands out because it doesn't seem to get as much benefit from that rejuvenation. So, whatever's going on in the blood, it's communicated to the heart. I think that's one of the organs, the liver, another organ. It doesn't seem to reach the brain. So you've got this sort of niche that is not reached by whatever's in the young blood. So, that's a nice niche to create a targeted therapy, right? Let's get to the brain. And of course, there's lots of scary things that happen to the brain with aging. So, that's where we are at the moment, but I think we've got plenty of flexibility as we move forwards to take on information. And, there's lots of people, there's no rule book at the moment, right? It's just, do you treat pets or is it, rejuvenate the reproductive system? Well, there's lots of ways to try and go about this. But the encouraging thing is that young blood has actually made great inroads for diseases of the brain, right? This is the amazing thing, even though the biological age isn't that much reduced. I think it's Tony Whisker with Alkahest, and there's something called the AMBER study. So in Parkinson's and Alzheimer's, you can actually affect endpoints in those diseases just with young blood, even though you're not affecting the biological age component very much. So, even when you're doing small things, you're affecting these diseases. Imagine if we brought the age of the brain down all the way, right? Like, the way that other organs are benefiting, like what that could do, right, as far as your risk of those diseases. So that's my inclination at the moment, but we don't need to cement that right now, but there will be a stage, like not actually that far in the future. The timelines on all of this, so I think 1 year to get to rejuvenation without Yamanaka factors,
Okay.
Another year to basically get a fully de-risked version, right? So we find what the nodes are, and these are like safe nodes, right? Like we can activate. So 2 years for that, and then sort of 3 to 4 years to go from like here's what we want to target to, you have to just go through the preclinical development gauntlet. So like model systems, toxicity, file IND filing, and ultimately when we want to, we're very absolutely deadly serious about doing this. We want to get all the way to dosing the first phase 1 trial. I guess the timelines are sort of 5 to 6 years. But that's, I'm a scientist, right? So this is borderline speculation. But we've got very, very, very recently a lot of expertise now providing their view, sort of informing these timelines a bit better. But I wouldn't worry too much about the time. It's about getting it right. It's better to have a fully de-risked therapy that goes the distance instead of rushing something through development that fails later, and that sets back everybody, right? Whenever $300 million goes up in smoke, and somebody else comes along with the real answer, yeah, they're less, it's a problem. So I think a bit of patience at the earliest stages actually pays dividends in the long run. But obviously we want these things as soon as possible. Not personally. I'm not confronting the abyss, but on the Shift team, for instance, Steve, who's my dad, he's much closer to these sorts of things, and he tells me from the front lines of aging, it's you wouldn't recommend it. You wouldn't recommend it, yeah.
Well, that sounds super exciting, and no, you're right. I think it's important to focus on the safety, but also you're right about also not cementing on a solid idea and just seeing where the information takes you. And I think that's the wise way of approaching it. And so speaking of just being in charge of running this company, also like me, you did biochemistry as training. How did you cope with becoming CEO of a company, and what kind of challenges do you face in this position?
Yeah, so it's quite a big switch from being an academic scientist to a CEO, and I think one of the things that I should have focused on in hindsight is communication. So when you come out of the lab, you're actually quite a scary creature that nobody you don't actually you can't really communicate with normal people. You can communicate in a robotic sense with other PhD students and things like that, but when you talk to other people, you can see their eyes glaze over. So the communication side, I sort of ignored this for a little bit, but quite quickly it was, you need to be a bridge builder, very quickly, in that position when you're sort of trying to bring together, say, capital and your ideas and stuff like that. I'm very fortunate that there's an entrepreneurial spirit in the family. My dad's doing this, and this might be more normal to me than it would to be to somebody else. So I could just sort of just ask him a bunch of questions. But yeah, communication was that was the playing catch up, right? Actually communicating properly with normal people. Not nobody. There's no such thing as normal, but just other people that could help us. And we were fortunate. Our first investor was a scientist first, right? So he was a scientist and then became an entrepreneur. So I could sort of talk to him as a scientist. I didn't really have to worry too much about talking to him and getting that investment. But then later on, we had to involve others. Sort of this communication issue. And the thing is, we stayed as a very small team for quite a long time. So when we set up in 2017, it was just two of us. I think we expanded to only 3 people. And it was only in 2021 where we doubled the size of the team. It's still not a large team.
Yeah, still pretty small.
Still pretty small, but it's still, it's a big change very quickly. And then communication becomes even more important. That's both internally and then trying to talk to people on the outside, and it's basically storytelling. I think I'm just going to recycle a bunch of advice I've been given, but I think there was a survey. It's what do you appreciate most from a pitch? And it's is it data? Is it story plus data, or is it just the story? And most people vote story. They sort of want to opt into the detail. They don't want you to download your entire brain on top of them, because they'll just they just don't even know. They can't swim in that, right? They drown. So if eventually I just told the story, and that was enough, and you could everything sort of flowed much easier. But I'm a first-time founder, so I'm sort of learning on the fly. I don't feel like I'm in a position to give advice, apart from the fact that we're still going, and it's going really well. And long may it last. But yeah, I think communication is the big one.
And you're still thinking about trying to grow the team bigger, or?
Absolutely, yeah. So all of a sudden, our bottleneck was financial. So we had the whole plan in place, and we just needed to go, right. We needed to just order a bunch of genes, do all this stuff, get equipment, and we've been able to close our round relatively recently, and now it's recruitment, right? We've got the fuel. It's a great problem to have, right? You've actually fueled to complete the mission that you set out to complete. We feel like we're very fortunate to have raised the money. So not all companies raise the money, so we're very fortunate to be in that category that did. Actually, the much harder task is then taking that money and executing on the plan. That's an even smaller number of companies, right? There's plenty of contenders, but then who actually gets there? So now's the really hard bit, right? Actually getting down into the really difficult and boring and dirty day-to-day of molecular biology. And if you've done a PhD, you just understand how hard it is to get things
Yeah.
to work, and how tedious things are, and how unrewarding. Like, you basically have to live with the fact that you're not going to get any like positive feedback. Like, you're not going to get any reward from your experiments. Or you basically have to just like get over it. Yeah, like, you don't get to tell the biology what it should do, or persuade it, or anything like that. And like, if you're lucky as a PhD student, in the 4th year you've actually got something to show for all that effort. There's many PhD students never, the biology never reveals itself. Yeah, and it's very character building, but it's this harsh reality that I think, that you need to sort of build that into your system before you get too excited about what might come in the future. You've got these very hard things you've got to do first. So I think everybody in our team, we can all go into cell culture if necessary and do the dirty and boring work, sort of stand in for each other. So I don't know if that's unique in the field or not, but I like to think that gives us a credibility, or we can all stare at the cold face of this, because this is a big problem we're confronting. Right? It's not, we can't wish it away with taglines and things like that. It's rigorous application of processes and techniques and things like that.
Yeah, no, I think teamwork is essential in science, and just being on the same wavelength with people and helping each other out when you need it.
Yeah.
And I guess that kind of makes you unique, being small and everyone knows each other really well, and I guess it kind of fosters that kind of relationship within the team. Yeah, this is
the useful side of a small team. You become like this special forces unit, where you can finish each other's sentences. Everything just flows very quickly. And you do lose that a bit with scale, so it's not necessarily more is more. Yeah, I think at the stage we are right now, I think we're actually a fantastic team to make this next step. The moment we reach, say, rejuvenation without aromatic factors, it becomes a much bigger undertaking that requires lots of people and a lot of expertise we don't have currently. But we're only ready for that once we've got that first signal or light from these experiments.
Yeah. So, I mean, as you kind of mentioned, there are like other companies that are also interested in like cellular reprogramming. What are your thoughts on it? Do you think there's plenty to research and it's like not a big deal, or just excited to see like other people in the space?
I think it's huge validation for this space, because we've been interested for a long time, but now you've got people that I guess the difference is the type of people that are talking about this now. They've got a much larger audience, right? And the audience includes like all of the best venture capital firms. Like, it's coming from the right mouth. Like, it's not enough for me to say this is real, we should do this, right? But when it comes from somebody else, actually, it's a huge sort of it's like the tide that lifts all boats, right? And I think basically I think this is a very good thing. There's plenty of room on the front lines, right? Basically, what we've got in front of us is infinite failure space where we're trying to find this biology, right? Like, it's not, you can't just roll the dice over and over again, and hope you arrive at the answer, because failure space is infinite and the success space is tiny. So I think as many approaches as possible is a good thing. And we're very focused on creating a vehicle to explore this huge space, as opposed to just firing things off in different directions and hoping it hits. So basically build the vehicle that allows you to pilot towards or away from, obviously towards the biology. So it's Nalbarian talks about emergent discovery and all these sorts of things. But yeah, I think it's a very exciting time right now. Like right now, I've sort of, because we've been going with Shift since 2017, and just the intensity of like developments, publications, sort of excitement, like mainstream excitement, has just ticked up in 2021 in particular. And I think this year, at the beginning of this year, we had this all hands meeting where it was like, all we've got to do this year is focus. It's a cliche, eye of the tiger. There's this whole thing going on around us, right? All we need to do is put one step in front of the other, and everything else follows. Because if the fundamentals aren't moving forwards, right, this whole thing can go away as fast as it arrived. But if the fundamentals move forwards, everything else is good. So you've got to ruthlessly focus on that. It's actually incredibly hard to do that day to day when all these fantastic things are happening around you. Yeah. And obviously we're in proximity to that. Right? There's going to be a Cambridge Altos Institute, and our advisor is now going to be part of that, and it's a fantastic thing, but it's also a defocuser. So we're trying to, how do we stay focused? And I guess as scientists, this is a great thing about our team. We're all scientists, and our source of energy is exploring the unknown. Like finding things out for the first time. So we built this vehicle, and we get to all get on board this vehicle and see things for the first time. And I think it's going to be this like natural focuser, like where we basically, we just perhaps go away for a little bit, or whatever you want to call it, stealth. We just go in because we're just sort of driving our vehicle and having a great time, and then ideally landing it on some safe rejuvenation biology, and then when we're there, everything else is good, and we can do what we need to do at that stage. So for us, this is the critical step right now, but it's a fantastic time, and I would encourage people to get involved. Don't see like a big company landing in the space as like a, as a warning not to get involved. It should sort of speak volumes to the solidity of the science, right, and the credibility of the field and things like that. I think more minds on this is a really good thing.
Yeah, no, I completely agree. And one of the reasons why I am still a scientist is I like being the first one to see the unknown, something that no one else has seen before. This is super exciting. And so I guess despite your extreme focus, do you have other interests in different areas of aging biology at the moment, or?
Yeah, so I'd say young blood-based approaches. So changing blood plasma composition. It's as far as translatability is concerned, it's amazing, right? Because you're just changing the levels of natural factors, right, in the blood, and in model systems you can achieve 50% rejuvenation. It's not 50.
Okay.
That's a big chunk of rejuvenation with something that's inherently translatable, and it's fascinating. I think again, Tony Whiskray, he did a study using single cell transcriptome, sort of machines. So he was trying to find out what is the functional pathway that's upregulated by young blood, right? So these mice are benefiting, but as far as the cells are concerned, what's happening? It's actually a very surprising result, and it's a very focal result, which was mitochondrial genes go up, and again, it's I'm trying to get away from being pigeonholed as a mitochondrial biologist, right? Because that didn't serve us well in the beginning, but it just keeps cropping up. So in young blood, you see the mitochondrial genes, we see them on. I'm not saying that that's the only part of the story. There's plenty of other interesting things going on, but I guess it's a nice connection to what I did previously. Mitochondria, there's so many different things you can study. They fuse to each other. It's this whole dynamics.
That's super interesting.
There's dynamics. There's the genome. Most of the genome actually migrated to the nucleus. So most of the genes are actually built outside the mitochondria and imported. So you still have this tiny genome where anyway, there's so many facets. It's like Narnia. You could just go in there and get lost and never come out. Which is you could I'll make the argument that's perhaps why I was blinking at the beginning. So you either love mitochondria or you know nothing about it. So there's very few people in the middle ground. But I'd say young blood's very, very interesting, and I think that will be the first type of therapy to cross the finishing line, as in a rejuvenation mechanism intervention that is clinically approved, not for aging, it'd be for something else like a specific age-linked disease, but be the first rejuvenation mechanism drug. To cross the finishing line. I might be wrong, but I think that will be it, and that will do a lot of good for other therapies that will follow. Because the moment somebody crosses the finishing line, then it becomes about effect size. Where do you get effect size? You need to know what the biology is. How do you know what the biology is? That's what we're working on. So at the other side of this, there's this huge flourishing industry, right? Once you can sort of rejuvenate cells and do these different things, it just becomes this brand new massive industry in all different directions. There's so much, it's not a zero-sum thing. It's there's space. It's like infinite space, right? It's up to you how far you can go with what you're doing. And I think if you're smart and you're excited about this, get involved as soon as possible.
Well, I think that's a great way to try and summarize this discussion up. It's been great talking to you today and learning more about Shift and its mission and how it's approaching the different questions it's trying to ask. And so I guess just a final thing, where can people go to find more information about you and to reach out and hear more about Shift?
Sure. So go to our website, it's just shiftbioscience.com. I also gave a talk to the Foresight Institute Longevity Group that goes into a bit more detail, and that might be fun to watch. It's a fairly lengthy video, but there's a bit more of our story there with some visuals. And we are recruiting at the moment for 3 priority positions. So my email address is daniel@shiftbioscience.com. So that's just a bottleneck for us. So sorry to be a little bit greedy there, but that would really help us. You can edit that out if you want.
No, the floor's yours. No, it's been great talking to you today. I just really appreciate the time you've taken to speak with me. I'm sure my viewers have found it very interesting as well.
Very welcome. Very welcome.
So I hope you've learned something in this video. Thank you to my Patreon supporters, and thank you for listening.